Alrighty, it's 2 o'clock, so let's get started. Hello everybody, I'm Jonas and welcome to the 5th talk of the online seminar series of population genetics. I'm just going to give a few words of who we are, in case you don't know by now. So we are a joint effort between the Population Genetics Group at University of Copenhagen and the Globe Institute. We are a network doing population genetics in Copenhagen, and we are usually doing monthly seminars physically, which we cannot do now. We have like scientific discussion and some talks, and then we usually also have some pizzas. So if you're interested in joining us, you can just follow this link here, or you can go to our website where we also have more information of the last talk and so on, in case you want to know more. And so far, this has been our schedule, and today we're at the 5th talk with Lucy. Yes, so today's speaker is Lucy. She did her PhD at UCL with Garrett Hellenhol, and David Balding and Mark Thomas. And then she continued to do a postdoc at also UCL with Francois Ballot. And now she's a senior research fellow in microbial genomics also at UCL. And Lucy has done a lot of studies in population structure and demography in African populations. She's also been working on evaluating population structure. And she's also worked a lot on pathogen evolution in population genetics. And today she's going to be talking about a very relevant topic in these times. And if you want to ask questions, you can either write in the Zoom chat or in the Twitch chat, which we should also be online on. Or you can wait till after the talk where you can raise your hand, or you can still write questions in the chat. And with that, I would love to get the word to Lucy. Yes.
Okay. Hi, everyone. Just managed to unmute myself. Just share the screen. Okay, I'm hoping that that looks good for everybody. Please notify her in the chat if anything doesn't look great. So thanks, Jonas, for the introduction. I'm a little bit sad not to be in Copenhagen this summer, but it's nice to have the opportunity to present to you today. So as mentioned, I work in UCL Genetics Institute, and my research is really focused on using phylogenetics and population genetic techniques to reconstruct the histories of humans and human-associated pathogens. And everything I do really makes use of large-scale genomic databases or data sets, and I really now focus on working to reconstruct the factors that have contributed to disease emergence, its transmission, and also its spread. And so most of my recent work, at least in pathogen genomics, is focused on bacteria, and particularly the agents of tuberculosis, Mycobacterium tuberculosis, and also many different classes of multi-drug resistant bacteria. But since kind of the start of the COVID-19 pandemic, myself and the group I work with have kind of focused some of our research efforts into understanding SARS-CoV-2, the virus responsible for COVID-19. And so I thought it'd be nice to take the opportunity today to present some of that work, and some of it is kind of ongoing. And really to give you a bit of background in terms of the genomics of COVID-19, how we can use kind of genetic data to learn more about this ongoing pandemic, and then to kind of present some of our own research and our own findings as we try and sort of keep up with the virus during this pandemic. So by way of background, and this is a story we're all now very familiar with, at the end of 2019, a cluster of pneumonia, thought to be viral pneumonia cases of unknown origin, were identified in Wuhan in China. And cases with this same kind of etiology started increasing. They spread rapidly and resulted in an epidemic throughout China. And this was followed by obviously increasing number of cases in many regions around the world. And by February of this year, the World Health Organization designated that this was a brand new disease, COVID-19, which stands for Coronavirus Disease 2019, after the year it was identified. And the virus that causes COVID-19 is severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2 as I'll refer to it now. And so by the 11th of March of this year, there was really sustained human-to-human transmission of this SARS-CoV-2 virus, and a global pandemic was declared by the World Health Organization. So I guess in this talk, I'm going to start by giving you a bit of a summary about what coronaviruses are and some kind of postulated origin scenarios for SARS-CoV-2. I'll then kind of move on to talk about the virus itself, this one species, and look at how genomic diversity has really emerged over the course of the pandemic, looking at traits such as population structure and also timings and divergence date estimates. And then finally, have a look at really the landscape of mutations that have accumulated in SARS-CoV-2 over the course of the pandemic, and suggest ways in which we might be able to phylogenetically infer their kind of plausible impacts. So I'll start with coronaviruses. Coronaviruses, and we've heard many talks about the coronavirus, but in fact they refer to very many viruses. They're a large family of viruses. They include those that cause the common cold, but also those that are responsible for more severe diseases. So for example, the SARS epidemic in 2002, 2003, and MERS, which first appeared in Jordan in 2012. They all look a little bit like this cartoon. I don't know if you can see my pointer, maybe you can. In that they have these kind of spikes around the viral surface, and this kind of spikes gives it a crown-like structure, or corona, which is where coronaviruses get their name. And coronaviruses can be split into kind of four main viral genera: the alpha, beta, gamma, and delta coronaviruses. And I've got kind of a quick and dirty neighbor joining tree here to give you some idea of the landscape of diversity we see in these viruses. So SARS-CoV-2, this novel coronavirus responsible for COVID-19, is now the 7th coronavirus that we know about that infects humans. So it falls with, for example, SARS and MERS as causing really quite severe respiratory disease. But other coronaviruses also include endemic coronaviruses. So we have alpha coronaviruses such as 229E and NL63. And other beta coronaviruses, HKU1 and OC43. And these 4 endemic coronaviruses are responsible for maybe 15 to 30% of common colds. So they're viruses that we're exposed to quite commonly. And in fact, there's some suggestions that there might be some cross immunity conferred by these endemic coronaviruses. But although there's 7 coronaviruses that are fairly common in humans, they're found in a very wide diversity of animal hosts. So here's the same tree now annotated by different host types, and you can see that essentially the coronaviruses are not fussy. They're found in a really wide range of hosts. So we've got deers, ferrets, giraffes, hedgehogs, all sorts really, as well as of course human infections. And this also includes kind of avian coronaviruses. So it's one important point to note that there really is a very large animal reservoir of these coronaviruses, or the coronavirus family. And SARS-CoV-2 is classified as a beta coronavirus. It actually falls within what's called the B lineage, or the sarbecaviruses, which is a subset of this genre. And so the first genome sequence of SARS-CoV-2, which is named Wuhan-Hu1, was collected in Wuhan province on the 26th of December of last year. So it was generated, assembled, annotated, and submitted to NCBI by the 5th of January, where it was available for people to download and the research community to look at. So this really was incredibly quick following the identification of this as a new disease. And of course, instantly having, or very quickly having, a genome from SARS-CoV-2 allows it to be placed into this family of sarbecaviruses, and subsequently questions to be asked about its relationship to other coronaviruses, including those that infect humans and also those that are infecting animal strains. So here's a phylogenetic tree, which is now just zooming in the sarbecaviruses, and there's a reasonable number of genomes here. SARS-CoV-2 is found here in green, if you can see, and then here we have the SARS-1 virus responsible for the SARS epidemics of 2002, 2003. So these are actually, if you look at this tree, quite different from each other. This corresponds to about 80% similarity. You'll see we've got all these other kind of blue sarbecaviruses, and these are viruses that have been isolated from animals, and in particular very many that have been isolated from bats, in particular one genus of bats, the Rhinopolis genus of bats. but there are some bat viruses from other species. And so it's quite clear that the sarbecoviruses themselves circulate quite widely in bats, but not exclusively in bats. So down here, I've got some kind of very unusual mammals, the pangolins, where there's been a good handful of sarbecoviruses isolated from these particular mammals. And so if we look at the two genomes which are currently available, which fall kind of closest or basal to SARS-CoV-2 that we have currently circulating in human populations, there, these two here, they've got names ROTG13 and RMYNO2. Both of these are isolated from horseshoe bats. Here's a little picture. They've got these kind of nose leaves which give them this horseshoe name. And both of these genomes have been found to be maybe 93 to 96% identical to the virus that's infecting humans, SARS-CoV-2. So these are, of everything that's been sequenced, or certainly the whole genome sequences that are available, the closest viruses we currently know about, and they do obviously fall within this animal reservoir. However, if you actually estimate the divergence time between these bat sarbecoviruses and SARS-CoV-2, you find that really they're decades divergent. So some estimates suggest, around 40 to 70 years. Others, kind of based on just synonymous sites, say maybe 50 to 37 years. So it's quite clear that these two viruses, although they're the closest that we have currently, are not plausible representatives of the direct ancestor to SARS-CoV-2. There's something a little bit more distant. So that leaves us with some kind of possible suggestions as to the plausible origins of SARS-CoV-2, and it's very likely that we have a sarbecovirus clade ancestor, possibly or likely of bat origin, which undergoes some kind of host selective pressure. There is some evidence in the genomes of kind of compositional biases consistent with host effects. We then have some recombination, and then the kind of landscape of diversity I just showed. So we see these SARS-like viruses that are existing in pangolins, they're existing in bats, and they're quite plausibly existing in a very large diversity of different species that perhaps we've not sampled and perhaps we're missing from our current genomic data sets. And so really the current working model is that we have a transmission from a zoonotic reservoir into the human population, and it really remains an open question whether this was from a bat-like reservoir or whether it was something else that we've still not sampled. Given it's quite clear that sarbecoviruses are circulating very widely in bats, it's not implausible this is the likely reservoir. But I think it's fairly important to keep a somewhat open mind, and bats do tend to get quite a lot of blame for infectious diseases. And this is a plot which comes from a quite recent paper in molecular ecology looking at the proportion of zoonotic viruses and what hosts they associate with. And in fact, rather than bats, we find that both rodents and primates carry on average a higher proportion of zoonotic viruses than we observe in bats. So it may well be that we identify at some point through large-scale genomic screening a different intermediate host. But the other point to keep in mind is that SARS-CoV-2 isn't just in human populations. In fact, it also infects or has potential to infect very many different animals. Right now, we currently have SARS-CoV-2 genomes that have been isolated from minks, from tigers, from dogs, and from cats, from kind of natural infection settings. And it seems that there's a vast array of animals that can potentially be infected if you kind of experimentally try and infect them in a lab. And particularly in minks, it seems the virus does quite well, and this is transmitting a lot between different mink populations, and this has led to kind of mass culling of minks at farms in the Netherlands. And there's also been efforts to kind of computationally predict the range of animals that SARS-CoV-2 could infect, and that's based on the relationship between this spike protein, this protein that gives this crown-like appearance to the virus, and the receptor. So in human cells, we have the receptor ACE2, the angiotensin converting enzyme. And this really suggests that there's a real large number of mammals for which SARS-CoV-2 could potentially infect. And so monitoring this animal reservoir and assessing the risk of these as potentially aiding transmission in human or animal populations is something that's quite an important area of ongoing work. So I'm now going to move in to just focus on this particular species of virus, SARS-CoV-2, and look in particular how we can use genomic data sets to track the emergence of genomic diversity over the course of the pandemic, it's worth noting that this is really an extensively sequenced virus, and really I don't think there's anything else in which we have kind of genetic observations at such kind of tight time points, right at the point where we have a virus that's kind of moved into a new host, in this case the human population. So what does the SARS-CoV-2 genome look like? Well, it's actually quite big for an RNA virus. It's about 30 kb or so in length. It has a genome structure a bit like this, made up of different open reading frames, some of which overlap. This genome structure is really quite consistent with that that's seen across the other coronaviruses. So you'll see that like the bulk of the genome, maybe 2/3 of the genome, is taken up by this ORF1ab, this open reading frame, which is responsible for encoding around about 16 non-structural polyproteins. This is a gene which is found across the coronaviruses. And then a decent chunk of the genome is then made up of genes that are encoding the structural protein. So this includes the spike, which is in red here and responsible for these kind of like spikes on the virus surface, but also other structural proteins, including the envelope protein, the membrane protein, and the nuclear capsid protein. And then we have like a small array of kind of quite small open reading frames, which are all kind of considered the accessory proteins. In SARS-CoV-2, we have 3A, 6, 7A, 7B, 8, and 10. And these will vary between different coronaviruses. So they're worth considering in terms of understanding kind of host modulation effects. And really, since the publication of the first genome, this Wuhan Hu1, back in early January of this year, there's now very many genomes available, and this has allowed large-scale genomic comparisons of this genome, of this 30 kb genome, during the course of the ongoing COVID-19 pandemic. And I think it's really worth putting this in perspective. I think in terms of a genomic response to an infectious disease, we're really in quite new territory. This is an example back from 2009, where we were facing swine flu caused by H1N1, which is a novel strain of influenza A. And this was really at the time the first epidemic for which any genetic sequence data was being generated in sort of close to real time. And so in about 29th of April 2009, the World Health Organization announced an alert level of phase 5. And 2 months later, this paper was published. It came out in Science, and it analyzed at that point the collection of influenza A H1N1 virus genomes that were available, or I should say partial genome sequences. So at this point, there were 11 partial hemagglutinin gene sequences available for analysis. And this really is a striking comparison to COVID-19 here in 2020, where on the point of announcing that we had a global pandemic, there were nearly 400 whole genome sequences available. And at the same stage, so a few months later, there were well over 10,000. So hopefully this little video works. So the speed of generation and sharing of SARS-CoV-2 genomes has been unprecedented. And this is really thanks to many research labs around the world generating data and sharing this genomic data prior to publication.
Interrupt you?
Is it not working?
Your slides are frozen.
No.
Can you maybe
reload it?
Yeah.
Frozen completely.
They're frozen in the ORF, the different genes slide.
Oh, no. Okay. Oh, so you missed all of the swine flu. Okay, I'll relaunch the show. Let me just give 1 second. How's that? Enjoy?
Let me just stop your screen sharing again. Oh, there we go. Yeah.
Yep. Okay, just yell at me again if it pauses. So this, I guess you've heard what I was saying, but yeah, so at this point in the swine flu pandemic, there were 11 partial hemagglutinin gene sequences. And now I'm going to move on to the video, which may well crash it again. I hope you can see this slide. We'll see. Hopefully you're all right with that. So this is really a timeline that illustrates the amount of genomes being uploaded since early January of this year. And as I sort of said, the speed of these genomes being generated and shared is really quite astonishing. This is by far the kind of largest genome repository or data set that I've worked with. And many of these genomes are available through a platform called GISAID, which I've acknowledged here. And so you can see that really there's been labs all around the world uploading and submitting data. And at this point, so this is made from today, there's about 55,000 or so genomes that are available, and these comprise 96 countries. So it's been an absolutely enormous effort, and it means that we have a really extensive data set in terms of understanding the genomics of SARS-CoV-2. And as you can acknowledge, you'll see on this map, this big blue blob, and this is genomes generated from within the UK, and actually has been kind of an astonishing effort here in the UK, with now over 25,000 of these genomes coming from the UK, and that's largely thanks to this COG UK Genome Sequencing Consortium, which also make their genomes available through this GISAID platform. So it's a pretty phenomenal resource, but it's not without problems, as is so often the case. So the first point to note, and these are all quite important points, is that these are all genome assemblies. So they're not raw sequencing reads, they're assemblies which labs have used their own protocols to sequence, sometimes using different sequencing technologies, assemble and then make available. So they've not necessarily all been treated the same, and it's difficult in most cases to go back to raw sequencing reads and actually check, for example, the allelic diversity at any one position. And so actually of this 55,000, there are genomes which are incomplete, that are too short, that are the wrong length. There's some with kind of marked or flagged up as low quality with high numbers of missing or ambiguous sites. And then aside from the genomic data, there's also you know, metadata associated with each of these samples, and on the whole, it's really good. We know where the samples were collected. We know to the day when the samples were taken from a patient infected with COVID-19. But what we don't always have is, for example, the travel history of the patient, so whether they've recently been abroad, for example. And it's fairly rare, although we have generally the gender and the age of the patient, we very rarely have any information on patient outcomes, so how they actually fared. But nonetheless, this is a massive data set, and it's a fairly well sampled and broad data set. And we can use this to ask questions about how the genomic diversity of SARS-CoV-2 has emerged over the course of the pandemic. And so all the results I'm going to present here are based on a data download that we've sort of been able to process and work on from the 4th of June, so this month. But of course, this is just a snapshot in time of a very quickly changing situation. Right now, that means that we're working with a data set of just over 23,000 genomes, so it's still plenty of data to keep us very busy. So here's a phylogenetic tree constructed over these 23,000 or so genomes. You can see that it's pretty big. This comprises just over 8,000 variable positions or mutations. You can see from the x-axis, which provides the number of mutations accumulated from the root, and the root here is the first genome that was sequenced, which arguably we could talk about whether better root should be chosen, but this is the first genome that we have. You can see that none really deviate more than, say, 27 or so mutations from the reference genome. So this really isn't very much diversity to be handling. It really highlights the fact that this is a young virus. It really hasn't had much time to acquire mutations since a common ancestor. And we can also annotate this tree by the location in the world where these genomes were sampled. And here I've just used kind of a continental region. So this is across 96 different countries. And we can use the pattern or the distribution of the place of the site of sampling to make some inferences about the spread of SARS-CoV-2. And one thing to note, and it's been mentioned, that I've previously worked in population structure. It's not uncommon, or very often I'm looking at data sets such as these and I see a strong geographic signal. So I see that genomes sampled in the same place look more similar to other genomes sampled from the same place. And really, in this case, there's quite a few colors across this tree, and it highlights the fact that there's a reasonable amount of diversity, despite there being a low number of SNPs to try and make sense of. And this is really quite consistent with the idea that we're all quite aware of, that there's been very many introductions of SARS-CoV-2 as a virus to geographic regions around the world. So to make that point a little bit more clearly, we can, for example, subset or color this tree just by genomes that have been generated within the United Kingdom. So here that's about 7,600 or so genomes in this data set. And you can see that really, if I was to take a random draw of this tree, like a genome from the UK could largely be found anywhere across it. So one genome really recapitulates or has the potential to recapitulate the global genetic diversity. And this is, as I said, consistent with very many introductions into the UK, where we have extensive genome sequencing, and highlights the fact that, well, one clear point is that there's no one such patient zero. You may have had the same thing in Copenhagen, but certainly here we've had many claims that Bob went skiing in January and was the first patient. And really this highlights that there's been so many introductions happening at so many different times that this concept doesn't really have any scientific value. And recently a paper came out maybe 2 weeks ago, or a post came out 2 weeks ago, that aimed to detect the number of independent introductions leading to transmission lineages within the UK. And the suggestion there is that there's been at least 1,300 of these, and that's really very much a minimum estimate. But the UK isn't unusual. So if we look, for example, in the US, we see a picture which is really quite similar. Also in Australia, where there's been large-scale sequencing efforts as well, we see that the genome's really full across the tree. And we can also look at, for example, Iceland, and I've highlighted Iceland because here there's been very extensive kind of contact tracing and population screening for SARS-CoV-2, meaning these 330 genomes are fairly representative. They're not just from very sick patients, they also include kind of healthier carriers. And you can see again that we have this situation where the genomes are found across the tree. And at this point in this paper, at least, there was an estimate of 42 introductions, but that was based on data downloaded in the 1st of April. And so kind of the model that we're really thinking about in terms of understanding the diversity of SARS-CoV-2 is that everything is everywhere. If I take any two viral sequences at random, on average, they are maybe 10 SNPs apart. So really quite similar, which means there's not much evidence to support claims of distinct lineages in circulation, at least at this stage. To just sort of illustrate that once more, if we take genomes from each of these continental regions at random, and this is like 100 sampled at random from each continent, and just look at the pairwise SNP differences, you can see that it's fairly low, and these all overlap. The one place where this is maybe less true is within China. So this phylogenetic tree now is just highlighting the Chinese strains, and you can see that they fall kind of predominantly into these two basal clades. And this really could reflect the fact that there was kind of sequencing in China where we suspect the virus or think the virus emerged first, followed by a lockdown. So the kind of genetic diversity of the virus is a bit more constrained, or it could simply be a temporal signal. So these were the first genomes to be generated. Some of you may well be aware that there's now kind of resurgence or outbreaks that have been underway in Beijing, and I don't quite have the tree ready, which includes some of these samples. But this is the tree that's been produced by NextStrain, which is a website which has really been incredibly influential to our work, and also been pretty amazing in terms of making transmission dynamics of SARS-CoV-2 readily queryable and accessible on their website. And they've included here the samples from the latest kind of Beijing outbreak. So we have 2 here and 1 here. So one point to note is that these fall within the diversity of what we see for the rest of SARS-CoV-2. So we can say categorically this isn't a new jump into the human population. And then the closest relatives in this tree, you can see, well, here's one in Denmark, but we've got genomes in Czech Republic, Russia, Spain, and Portugal. So it seems like one possible scenario is that there was an introduction from Europe to China of this outbreak, but this is quite challenging to say. And I say that because if you look at these genomes, they're from all across Europe, and we've seen that many genomes can be in many different countries, so we can't really nail down where. And you can also see that these genomes have accumulated some mutations, so it's quite likely that there was some kind of circulation going on in Beijing before this outbreak was actually identified. But nonetheless, this kind of highlights how you can use patterns of genomic diversity to make insights that might be epidemiologically relevant. And kind of talking about epidemiological relevance, one really big question that keeps coming up again and again and again in the context of COVID-19 is, when did it start? And this is obviously important to address, firstly because we'd like to know how long it's been infecting humans, so how much baseline immunity might we expect in the human population? Is it something recent, or perhaps a virus that was circulating for a really long time and then suddenly became more virulent, for example? but also because this is a parameter which is quite fundamental to some of the epidemiological models that are being used to inform some of the public health making decisions. So is this an older virus, or is this something that's really quite recent? And of course, there's very many anecdotes, and I have this as well from friends and family who claim to have had coronavirus-like symptoms quite early on in the year, or perhaps earlier on last year. And so knowing the origin is something that we do care about. Genomic approaches and phylogenetic approaches do allow us to make an estimate of this origin, in particular by taking an alignment or phylogenetic tree like this and making a prediction as to when all of these viral sequences last shared a common ancestor. And so this is based on the premise that we have a relationship between the amount of genetic divergence that accumulates from an ancestor, in this case the root, compared to the time at which a virus was sampled from an infected patient. And we'd expect, given the principles of evolution, that we would see a significant correlation between the time of sampling and this root to tip phylogenetic or genetic distance. And indeed, that is what we see for SARS-CoV-2. So here's the regression here. And it follows that the intercept of this regression provides kind of a cursory estimate as to when all of these strains last shared a common ancestor. So in this case, you can see that we estimate this to 2019.86. This corresponds to around about the 10th of November, which suggests kind of an emergence towards the end of last year. I should say this is really a cursory estimate, but we've performed a little bit more of an in-depth analysis using a package called tree data on a smaller subset of genomes, on 7,500 genomes, which gives us a window or a time frame of opportunity for this jump into human populations from about the 6th of October last year through to the 11th of December. And I should really point out that we're not the only ones doing this. So there's been many efforts to estimate this ancestor of SARS-CoV-2, and in particular using different methods. So for example, using Bayesian tip dating approaches, for example, in BEAST, which does require subset in the data set quite a lot because there's just so many data points at this point in time. But all of these are really very consistent. So this really points to this being quite a young virus. I've also put up this table because it highlights the substitution rates that have been estimated for SARS-CoV-2 based on these kind of approaches. And firstly, they're all fairly consistent, but they're also very similar to those we observe in other coronaviruses. So there's nothing to suggest that this virus is mutating more quickly or more slowly than we might expect, given the family of viruses it belongs to. And in fact, actually, if you look at RNA viruses, this coronavirus is really actually quite slow in how it mutates. And that's because it actually has the presence of a non-structural protein that acts as a proofreading mechanism for the RNA polymerase. So it actually tries to catch out some of these sporadic mutations which might appear during viral replication. So it seems quite clear that we have an emergence towards the end of last year in our estimates, and probably the best estimates of other colleagues working on this question is towards early or mid-November. And so this is not incompatible with reports that have been kind of coming out really quite recently of observations of SARS-CoV-2 here in Europe towards the end or very end of last year. So in particular, some suggestions of SARS-CoV-2 positive patient biobank samples in France in late December, and a recent medRxiv preprint which just came out looking at or detecting SARS-CoV-2 positive samples in wastewater in Italy around about the same time point. What it is incompatible with is some recent reports of SARS-CoV-2 positive samples in Barcelona towards the early part of last year, and I think really there's a wide consensus within the scientific community that this paper maybe had some problems, in particular the lack of a negative control. So it really seems that we have a emergence, we have a disease which is picked up reasonably quite, really quite quickly, and then we have a spread out of Asia and into Europe. Okay, so that's kind of some really inference we've been able to make, this concept that in terms of geographic diversity, you'd have to work quite hard to find it. Everything seems to be reasonably everywhere, and really this idea that we have a quite, or all support is for a recent origin of SARS-CoV-2. And I'm going to finish on a section which focuses in on these mutations and looks at the landscape of mutations in SARS-CoV-2, and in particular focus on recurrent mutations and what they might tell us about how the virus may or may not be changing over the course of the COVID-19 pandemic. And so we've seen how mutations can be used to inform us on how disease is spreading, and really highlighting the fact that this is a young virus. It's kind of naive to us as a human host. The amount of diversity is really quite small, 10 SNPs or so. So despite all that, there's really intense interest from the media, the public, and the scientific community on mutations as they've emerged in the SARS-CoV-2 pandemic. And we care about mutations, and in particular, we care about looking for regions or putative regions under selection. Mutations can also be useful, or the landscape of mutations can be useful in terms of inferring regions of the genome that might be more constrained or invariant, and these might be the regions that we'd want to target with drug and vaccine efforts to avoid kind of rapid vaccine evasion or drug resistance in SARS-CoV-2. And also there's quite intense focus on whether any changes or mutations in the virus have resulted in modulating the virulence or the transmissibility, and arguably picking up such changes early would be helpful in kind of controlling the pandemic. So mutations in SARS-CoV-2, or indeed any virus, are a result of or plausibly a result of a couple of factors. There may well be, or there will be, copying errors during viral replication. Mutations may also arise due to a combination between viral lineages co-infecting the same host, though as I mentioned, there really is not strong evidence for very different or genetically different lineages at this stage. Mutations can also be induced by host RNA editing systems, which form part of our own natural host immune response, and this is something I'll pick up again on later. And then finally, and this isn't a biological property, but certainly mutations that we observe within alignments of SARS-CoV-2 genomes may well be the result of artifacts, so sequencing and also assembly errors. And we've taken a few steps to try and catch and account for these. And most mutations, as they just accumulate, as the virus replicates and evolves, are expected to be very neutral. Any very highly deleterious mutations, so those that, for example, prevent the virus even infecting a cell, they're just going to disappear out of the population. Slightly deleterious mutations may be retained, at least for some period of time. And those mutations which reach high frequency are likely to be those that are either neutral or advantageous. And that's a scenario that we need to be able to distinguish. So we can screen along the genome, this 30 kb genome of SARS-CoV-2, and ask, well, where do we see mutations? And this is based on our data set of around about 23,000 genomes. The kind of landscape of mutations looks a bit like this. So you can see that we obviously have variability occurring genome-wide. All sites have potential to mutate, but there's also some which occur at really quite high frequency that we may want to follow up on. And I should say that our kind of current version of the alignment is available as a Shiny app, which you can query. And this was built by Mislav, who's a PhD student in the group, and he did kind of a quite amazing job given he'd never used Shiny before. So yeah, a little plug for him. So one challenge when we see these mutations, as I mentioned, is those that arise at or arrive at high frequency can be the result of neutral evolution, or they can be the result of adaptive changes. So it's quite important to be able to disentangle those mutations that arise from demographic processes. So for example, we have a virus that carries a mutation or a suite of mutations that's introduced early into a country, perhaps when there's not very many preventative measures in place. It spreads quickly and we observe that mutation at high frequency. Compared to a scenario where we might have mutations that reach a high frequency because they're adaptive. They actually play a role in changing or supporting the virus in its own evolution. And really our focus at this point in the pandemic, given the diversity and the number of genomes we have, is to really hone in on those mutations which we see have arisen repeatedly and independently, so homoplasies, given the phylogenetic tree of the virus. So I can return to this plot of SNPs along the genome when we actually find that around about 3,000 or so of these are homoplasies. And this is really a lot, and it really highlights the fact that these are data sets which large and well annotated, but also have some issues in terms of sequencing artifacts. And we really want to filter down this list of 3,000 or so homoplasies, which could arise, for example, from these sequencing artifacts, to those that we believe are the most plausible candidates and biologically plausible candidates for sites that might be under selection, for example, host adaptation or changes within the biology of the virus. And so we took a number of strategies, or chose a number of filters in order to try and get this to a kind of more manageable list. Firstly, we masked the ends of the alignment, and this is an approach which us and many others have been taking, because it's these ends of the alignment that are difficult to assemble using de novo assembly techniques, but are also most likely to be erroneously aligned following multi-sequence alignment. And we also masked out those sites that have already been identified as sequencing errors, and that's thanks to efforts of, for example, Nicola Di Maio and also NextStrain, who make that list publicly available. We also kind of reasoned that if we see a mutation that's a plausible sequencing error or homoplasy, it's very likely to show up, or it might well only show up once or twice across our entire data set. And so we relied on, or we made the assumption that we should observe homoplasies that occur in at least a decent fraction of our data set, in this case 0.1% of isolates. And we also reasoned that if we see these homoplasies, but they're kind of naive to their neighbors, they occur as singletons on the phylogenetic tree, then again, they may be more likely to be artifactual. And finally, our final filter was, we noticed when processing these plausible lists of homoplasies that there were some recurrent mutations that occurred only in genomes that had been uploaded by the same originating or submitting laboratory, suggesting there might be something in their sequencing or bioinformatics protocol which results in them always calling a SNP wrong. And so we also used a filter whereby we said we have to observe this position, this recurrent mutation, in more than one submitting or originating laboratory. And so that takes us down to a much more manageable list of 328 candidates, of which around about 60% were involved in non-synonymous changes. And I should say that we also validated some of these 328 by using, where available, short-read sequencing data. So applying kind of the more standard approach of doing alignment and quite stringent SNP calling to assess that we actually were genuinely calling variants at these positions. And so here's the scan along the genome again, but now I'm showing the homoplasic position. So you'll see that the y-axis is now the minimum number of changes on the phylogeny that's invoked by one of these mutations. And you can see that there's, again, a fairly even distribution across the genome, but there are some kind of hotspots or high sites. And in particular, this is one particularly, I guess, a mutation involved in a very large number of changes on the phylogeny. So it's convergently evolved multiple times. This is a position 11083, which is found within this big ORF1A open reading frame. And it encodes, or it's in part of the region of the gene encoding this polyprotein NSP6, which is one that's been implicated in kind of immunogenic reactivity in human cells. But you can kind of make stories up about any of these mutations, and certainly we've focused in quite strongly on, for example, the spike mutation. So this one here is position 21575. And that's because the spike is such an important protein in terms of modulating cell entry. There's also been several claims of mutations within the spike proteins being involved in increasing transmission, and one in particular, D614G, which has had a lot of press, that actually falls here within our data set. So although it is a homoplasic position, it doesn't induce very many changes on the tree. And so understanding what some of these 328 kind of candidate mutations do is obviously of relevance. Do they have any impact on the virus, or are they these mutations which are just at higher frequency, or because of demographic processes? And really, there's been some horror headlines in the context of SARS-CoV-2 genomics, claims of mutant strains that are kind of going to take over the world, or they're more contagious, aggressive, or they're going to really affect all our efforts to identify a vaccine target. And so really a crucial next step in our analysis is to take these 328 homoplasies and assess whether they have any impact on the biology of the virus. So for SARS-CoV-2, or kind of indeed any virus, transmission fitness can be considered as a proxy for the overall fitness of the virus. And this assumption allows you to make some phylogenetic tests of fitness effects. And that's based on the fact that we might expect a virus carrying a mutation that really helps it out to have very many more, or a larger fraction of descendants compared to one that doesn't. So here's a kind of a phylogenetic tree, and you can imagine I've kind of annotated this with a plausible homoplasy. This is the red nodes and tips on this phylogenetic tree. And under kind of random sampling, and you've seen that SARS-CoV-2 is a reasonably random sample. There's genomes that are really recapitulate the global diversity. We might expect that a mutation that positively affects a pathogen's transmission fitness will appear in proportionally more descendant nodes. So here we see that there's more red, for example, than white tips descending from this parent node. And so to test this more formally, we developed a phylogenetic index called the ratio of homoplasic offspring. We call it the ROHO score. And this really allowed us to perform these kind of analyses across the whole genome for all possible considered nodes. So here I've got another phylogenetic tree, and homoplasic positions or recurrent mutations are again annotated in red. And this tree has 3 possible parent nodes that we might consider for this ROHO statistic. So 1 here giving rise to these red tips and these white tips, 2 which again has the red and white, and 3 which has a single observation of the homoplasic allele and 1 observation of the reference allele. And we considered a set of criteria really to ensure that using these kind of differential statistics was robust and also symmetric, and by that I mean non-biased. So firstly, to avoid pseudo-replication, so to avoid counting the same node twice, we really required that the descendant node had no subsequent parent nodes, which then acquired the homoplasy. So just to make that clear, for example, for parent node 1, we see we have a secondary emergence of a parent node carrying the homoplasic site at 2, which means that we would discount 1 because we don't want to count this position twice. And also we found that one way of ensuring that our statistics were non-biased was to also only consider parent nodes, so one of these 1, 2 or 3s, where there's at least 2 descendant tips that have each allele type, so the reference and the homoplasic or alternate allele. And so on this basis, we would also exclude 3 because we only see 1 observation of the homoplasic allele and 1 observation of the reference allele. And this was to avoid kind of 1 over n or n over 1 type comparisons. However, this still leaves us with 1 parent node that we can test, in this case number 2 here, where we observe that we have 4 descendants carrying the homoplasic allele and 2 carrying the reference allele, which gives us a ROHO index or ROHO score of 2. And so we might expect that under completely neutral evolution, this ROHO score would be balanced, and as such a log 10 ROHO index would be equal to 0. And to check this was the case, we conducted a set of simulations that were designed to explore what patterns of ROHO scores we might expect under completely neutral evolution. So to start with, we simulated a set of discrete traits on our real phylogeny of SARS-CoV-2. So we said, let's randomly assign traits to the nodes on this tree, treat them as homoplasies, and calculate our ROHO scores. And this gives us a distribution like this. So remember, a ROHO score or log 10 ROHO score around 0 is kind of equal to what we might expect under neutral evolution. And you can see that here we have a collection of kind of false homoplasic sites that all fall within this neutral expectation. And the second thing we did was to genuinely sequence an alignment from scratch, mutating at the rate that we observe within our true SARS-CoV-2 phylogeny, and again to build trees, annotate homoplasies, and conduct the ROHO score. And again, you can see that our ROHO scores, based on the y-axis here of 0, really sit around this line of neutrality. So this suggests we do have the ability to detect neutral evolution well. So what do we observe in our real data set? Well, in our real data set, we found that we had sites which passed our filtering criteria for conducting a ROHO score and also appeared more than 3 times within our data set. So were replicated 3 times for 80 possible homoplasies. And for all of these, we conducted this ROHO scoring index to assess the impact of these recurrent mutations on the SARS-CoV-2 viral fitness. And here are the results. So you can see that very many of these sites across this 30 kb genome are falling in or around 0, albeit there's a bit more diversity than we saw in the simulations. But what this suggests to us and what we found is that none of these positions were significantly associated with either an increase or decrease from what we would expect under neutrality. And actually this real signal was incredibly neutral. So if we take the mean log 10 ROHO score, the median log 10 ROHO score, or even the fraction of observations that fall above or below this 0 line, we find that they're kind of symmetrically distributed. So this really seems to fit our expectation under neutral evolution, and suggests that to date, there's no recurrent mutations that are really significantly impacting the transmissibility or the fitness of SARS-CoV-2. And so we do, however, see very many mutations. And then one further question is, well, if these mutations aren't, if they're evolving neutrally, they're not having a strong impact, then do we see any interesting properties about them in terms of, for example, their base composition? And so we went on to explore each of the nucleotide changes at these SNPs and also at each homoplasy. And interestingly, doing this, we observed some really high compositional biases. So here are two plots. Here's all the SNPs in the genome, and here's our recurrent mutations or homoplasic sites. And the x-axis gives you possible transitions. So we have CTU, HG, GTU, and so on and so forth. And quite remarkably, you can see that there's this really high elevation in CTU changes, and actually 98% of mutations which involved a cytosine corresponded to these CTU changes. So this certainly seems an effect which is non-random. And so this suggests to us that mutations that we observe in the SARS-CoV-2 genome are fairly neutral, and they're not solely the result of errors by the viral RNA polymerase as it's making replicates during viral replication. And one kind of intriguing possibility, and it's one that I really didn't know very much about until we made this observation, is the possible action of human RNA editing systems. So these are systems which have been implicated in our own innate and adaptive immune response. And amongst the kind of most compelling candidates in this case of these RNA editing systems is the AID or ApoBec family of proteins. These are cytidine deaminase enzymes which catalyze this change from cytidines to uridines, so the C to U that we observe in RNA viruses. And this is not an observation that we've just made, and we were really informed by the work of Peter Simmons and also De Giorgio et al., who also observed that we see this elevation in C2U changes, possibly as a result of ApoBec family modulating. And this is a property which has also been observed in some of the other coronaviruses, so also in SARS and MERS. And it's also reasonably common, or a kind of base compositional change that's been observed within HIV and SIV. So it seems that these are important enzymes in terms of our own innate antiviral immunity. And so if we take our alignment of SARS-CoV-2 genomes and we look at all possible combinations of freemers, so free letter strings, we find that we do have an enrichment for strings with a letter C or cytidine in the middle of this free letter string. And the reason we did this is because ApoBec deaminases do preferentially targets particular motifs within the genome, and I've highlighted in arrows some of those. So certainly the ApoBec free A family of proteins seems to target preferentially CCA and TCT, trinucleotide changes, which we do see at a high rate within our data set. So it does seem that many mutations that we observe in SARS-CoV-2 may be driven by this host ApoBec-like genome editing, which I think is really interesting. Whether this has functional consequences is kind of yet to be shown. So it could be that our immune system is kind of heavily editing RNA viruses to maybe introduce stop codons or somehow leave them non-functional. It's also possible that by kind of increasing mutations or adding mutations into a virus, this could somehow fuel the virus evolution. So nonetheless, whatever impact this is happening, I think it's very worth being aware that we have this RNA editing system, which is certainly contributing to the emergence of genomic diversity that we're observing within SARS-CoV-2. So that kind of finishes up this section, which is really looking at the landscape of mutations and recurrent mutations and their inferred impact. So I'll finish with a summary. So large-scale genomic data, and it really is large-scale, from SARS-CoV-2 is offering us new opportunities to respond to this pandemic, so inferring the origin, the spread, the evolution of this coronavirus. In all likelihood, SARS-CoV-2 has a zoonotic origin. We think that it jumped into the human population in around November of last year, and then it circulated really rapidly into many regions, most regions around the world. And as a result, most countries, and I've highlighted particularly the UK, US, and Australia, but I'm sure it's a similar case for any other kind of densely sequenced region, many countries have experienced these many thousands of introductions. There's no one patient zero. And all these strains that are being introduced, there's subtle differences between them, but they're not, they're really genetically quite similar. And this suggests that to date, there's no phenotypically different lineages in circulation. Mutations are a natural outcome of virus replication, but they should be closely monitored. We're trying to monitor them closely, and one way of managing this is to focus on those that have emerged repeatedly and independently. But our kind of ROHO index suggests that to date, none of the recurrent mutation candidates that we tested has significantly altered the transmissibility of SARS-CoV-2. So it's not increasing its transmission, it's not decreasing its transmission. In fact, it seems to be behaving quite similarly. And finally, we observe these marked base asymmetries within the genome or the alignment of SARS-CoV-2, and this may well point to the role of human RNA editing mechanisms, which seem to be very important in the evolution of SARS-CoV-2. So I should just finish up by pointing out that this work is kind of available as a publication and also as a preprint that we're updating. It involves kind of a large team of colleagues who have really all contributed. I've tried to credit people during the slides because it's really been very much a team effort. And also I should point out that all of this is possible because of the huge amount of genomes that have been uploaded by GISAID. So I've been quite happy sat here in my living room analysing genomes that have been generated without having to do any of that hard work myself. So thanks very much for your attention, and I'm happy to take questions.
All right, thank you very much, Lucy, for a very interesting talk. Yeah, so there we will take questions for Lucy. So you can either write in the Zoom chat or raise your hand and we'll pick you, or you can write in the Twitch chat.
Thank you, Lucy. It was a fantastic talk.
Cool.
We have already 3 questions. So I'm going to start with Mohammad Tajub from Pakistan. He asks, like, if there is difficult places to access for samples of pangolin, how do we know that there are carriers of COVID? And if I can expand this question, can you comment anything about how much we know about the diversity of non-human carriers?
Yeah.
And what are the implications of not knowing possibly some of this diversity?
Yeah, so I've gone back to my tree. Hopefully that changed. So yeah, so the observation of kind of SARS-like coronaviruses in pangolins is kind of based on these. I think it's about 6 or 7 genomes. These aren't the SARS-CoV-2 virus, so they're really quite divergent. They're not the same virus, but they are reasonably close representatives. And so there's been some quite careful studies of whether these are a possible intermediate host. They seem a bit more divergent, so kind of a priori you might suggest that a bat reservoir might be more likely. There are some kind of intriguing properties. So certainly if you look at the receptor binding domain of the spike protein in the pangolin associated viruses, that's actually a little bit more similar to the receptor binding domain we have in SARS-CoV-2. So it's not implausible that we had some kind of history of a combination occurring within these SARS-like viruses before we get to the kind of plausible direct ancestor of SARS-CoV-2, which kind of remains unsampled. And I think knowing what that ancestor is, I think there's a great desire amongst many of us to understand the origins of SARS-CoV-2. I think as humans, we're kind of always intrigued by origin stories, but also, of course, it's important to understand which are our most vulnerable zoonotic reservoirs. So, where should we be monitoring? Where should we be looking if we're worried about subsequent host jumps of coronaviruses or indeed kind of other zoonotic pathogens?
Yes, thank you. So plenty to do in those fields.
Yeah.
Fernando, you can go ahead, please.
Hi, Lucy. That was a fascinating talk. I was curious about this, like, human RNA editing systems that you say there may be some evidence for. Like, I'm not that familiar with them. Like, what is adaptive in our response to them? Like, why would we want to create an elevation of CTU changes? Like, is it something about RNA stability?
Yeah, I don't actually know specifically, and I have to say this isn't my area either, but I don't know specifically about why it's CTU. This is the target. There seems to be characteristic motifs that are targeted by various RNA editing mechanisms, of which APOBEC is just one. And certainly the kind of motifs that we see elevated are those that relate to the APOBEC-free family of proteins. And I think the idea is that we have, an infecting virus, it's starting to replicate in human cells, and then at that point we have a system in place to induce these kind of hypermutations or CTU changes. And I guess the kind of working knowledge is that these act almost like an internal kind of therapy to try and just induce sporadic changes into the virus that are likely, if you induce enough through hypermutation, to be deleterious. And I think this is why we tend to see lots of homoplasies. We see these kind of recurrent changes and this elevation of the CTU. But so there have been talks, some people have kind of suggested that maybe if we can harness that potential and kind of increase or amplify the action of ApoBec, then that could be a useful therapy. But at the same time, we're also increasing the landscape of mutations of which selection can act on. So potentially it's also not a good idea. So I think it's really interesting quite how these things work and what they mean. Thanks.
We have another from the chat from Anders. Is there any evidence of recombination or co-infection found in the COVID-19 samples?
Yeah, so we haven't really detected strong evidence of recombination within SARS-CoV-2. Within the related coronaviruses, there does seem to be some evidence of recombination events occurring kind of in the direct ancestor. There was a study quite recently out of the BDI in Oxford where they've looked at within patient diversity, and there is some suggestion that there's kind of allelic repertoire that suggests that there's more than one strain potentially in circulation in patients. They haven't exactly nailed it as recombination, but what they have used is that relationship to the kind of allele spectrum that you get in an infection to map transmission, which is quite an interesting approach, because what we actually have is a virus that's evolving quite slowly. We're passing between patients, patients are passing to each other, and actually the genomic diversity is low. So there's very many duplicates in our data set, for example. And so maybe there's these subtle lineage effects that you can use to infer transmission, but whether that's actually in host or combination yet, I think we don't know. And part of that is because we don't have the raw reads and we don't have the number of patients kind of longitudinally sampled within host.
Yes. Thank you. So now Ida.
Yes, thank you. Thank you so much for the talk. It was really interesting. I guess I was just wondering if you could say a little bit more about your choice of root in your phylogenetic tree, and related to that, how certain you are about the place of origin?
Yeah, no, it's a good question. So essentially we don't have the ideal root. So, you would normally root phylogenetic trees by an outgroup, and you want that outgroup to be kind of equally genetically distant to everything you have, but also not too far away that actually the root can kind of flip-flop in different directions within the tree. And so one possible solution, I've got the slide up here, is to use, for example, this genome as your root. But actually what you find happens is that it's a kind of equivalent to midpoint rooting the tree. So you can't really resolve where your root should be based on that genome as an outgroup. And so it's a potentially non-perfect solution, but we've kind of stuck with what the consensus is in terms of what people are doing, which is saying, well, let's use our first observation, which is the Wuhan Hu1 reference genome. I mean, if we look at the kind of diversity of viruses we have in circulation, certainly those that are early in the outbreak that have acquired less mutations are those that are found in Asia. So I think the Asian origin is very confident, but we may well sequence something at some point from one of these intermediate hosts, which will serve as an outgroup, which might subtly change things, but I'd be surprised if it changed things very much.
Great, thanks.
Pleasure.
We have another one, but it's very similar question about routing. So yeah, if you tried other routings other than the Chinese sequence, but I guess already.
Yeah, we tried R8PG13. So we tried at the time it was the closest genome. The other back genome has come out subsequently. And it really didn't seem to make much of a difference, but this is, yeah, as I said, I think as new genomes come available, there may be possibilities to try out with different references. As I said, the first genome we have, Wuhan Hu1, everything is maybe at most 30 SNPs different from it. So the choice of reference and outgroup maybe won't make so much difference within what we currently have. I think it will be the addition of a new genome which would plausibly change things a little bit.
Do you have any other questions? Anyone raising the hand? Yeah, Cindy Santander. Go ahead.
Hi, Lucy. Thank you so much for that talk. It was super interesting. One of the questions that I had was in regards to treatments for COVID-19, and specifically knowing whether we're actually at a state where treatments would be able to target specific loci within the virus. So I think a lot of vaccines have kind of circulated around working with targeting spike but for example, protease inhibitors, like how good would we be able to actually target any region that was under adaptation in COVID-19?
Yeah, so I have to say this like falls outside of my remit a little bit, but I can at least comment that you know, the kind of amount of genomic diversity that we observe is this kind of landscape of mutations which aren't necessarily very constrained to particular regions of the virus. So I think efforts currently to target specific regions are not misplaced, because we don't see huge amounts of diversity, at least at this point. Most of the vaccine efforts are focusing on the spike protein, which I think is a reasonable target. This is the region of the genome that's being, or the protein that's being used to infect human cells. So it seems a good target, but not exclusively. And so right now, my understanding is there's about 100 vaccines or so that are kind of in the works, and the general success rate is maybe 6 to 10%. So I'm kind of hopeful that one of these will pan out. One thing I think would be useful, and I know others are kind of doing, is to use what we know about, for example, SARS, which we've had much more time to study. And there are regions using kind of comparative genomic approaches, which are essentially very, very similar between these two viruses. And so one possibility is to co-opt some of those treatments, which sort of got abandoned at the end of this kind of SARS epidemic, and pick them up again in terms of targeting SARS-CoV-2.
Thank you.
Thank you, Lucy. Maybe we have time for one more. Yeah, Anders, again, does your analysis support the notion that much of the infections are due to super spreaders?
No, I don't think it does. I have to say, I'm trying to think what we would expect in terms of the phylogeny, which might be like more of like a star-like phylogeny. I think in reality what we see is kind of this kind of accumulation of diversity, which is kind of almost fairly randomly distributed. So I would say no to super spreaders. One important thing to note is, and genomic databases are always imperfect, and this is certainly focusing efforts on sampling, or generally people who've ended up in hospital very, very sick. It's those genomes which are getting sampled. And so perhaps we don't have a very good understanding of what's going on in carrier populations, and potentially there will be these kind of pockets of spread that we're currently not really sequencing. But I think right now, I think it's more a kind of phylogenetic picture, kind of systemic, community and imported transmission events, as opposed to kind of one person, a big spread, and so on and so forth.
Thank you, Lucy. Okay, it's 3:07. So maybe there is one more, okay?
Okay.
Rasmus Hella. You can now unmute yourself. Yeah.
Yeah. Hi, Lucy. Thank you. Just in relation to your last answer, I think you mentioned that you have a lot of data from Iceland, where presumably they tested more widely and not only symptomatic cases.
Mm-hmm.
Did you have any chance to sort of contrast these results from a more broad testing regime with those coming mostly from very sick people, and whether there are any differences in the evolutionary patterns?
Yeah, so I haven't, and it would be nice, and this gets back to the point I sort of was kind of careful to make about metadata. So there generally isn't information on the patient's status, so it would be lovely to have these were, you know, very mild infections, and these were more severe infections. And this is the tree we have for the Icelandic genome. So the other point is, it's actually, you know, this tree is on 23,000, so we have 330. So I think even if there was an effect, we'd be quite underpowered to see it. At the same time, you can see that this kind of idea that we have a genome which can recapitulate any bit of the global tree does seem to hold true. So perhaps there isn't too much of an effect, but right now I think we need a few more genomes, to try these things out.
Okay, thank you.
Thanks.
Okay, that seems to be it. So thank you again, Lucy, for a fantastic talk.
Pleasure.
And everyone is welcome next Friday. Ana Safo Malaspinas is going to talk about human migrations from both ancient and modern DNA, 2 p.m. Central European time. And thanks again, and thanks everyone for joining.